is Professor James Moon is John Gideon Sirleaf Professor in the Department of Pharmaceutical Sciences and Biomedical Engineering at the University of Michigan, Ann Arbor. His research program aims to develop novel biomaterials-based strategies to advance fundamental understanding of the immune system, with the ultimate goal of improving patients' lives with effective vaccines and immunotherapies. He has published more than 100 research papers, including those in high-impact journals, including Nature Materials, Nature Medicine, Nature Nanotechnology, Nature Biomedical Engineering, Nature Reviews Materials, and Science Translation Medicine. He has 10 pending approved U.S. international patent applications, and his novel nanovaccine delivery technologies have led to 2 new startup companies. Dr. Moon's contribution to the field has been recognized with many awards, including AAPS Emerging Leader Award, NSF Career Award, DoD CDMRP Career Development Award. He obtained his bachelor's degree from the University of California, Berkeley, and his PhD from Rice University. Afterwards, he completed his postdoctoral training at MIT. The floor is yours, sir.
All right, thank you very much, Ahmed and Ali, for this kind introduction and invitation to speak here. I wish I could be there in person, but hopefully we can see each other in person pretty soon. So today we'll talk about what my lab has been working on the area of engineering strategies to modulate the gut microbiome and immune system. And I hope to get a good feedback from you to further improve our projects. So before we begin, this is my relevant disclosure slide. I serve as a co-founder and CSO in Evoke Therapeutics, which will be the first topic of this presentation. So my lab works on cancer immunotherapy and ways to improve immune checkpoint blockers. That's one of the major areas in our laboratory. This is showing patient survival curves in response to traditional chemoradiation that invariably lead to mortality. But with the invention of immune checkpoint blockers, now we have a subset of patients that are considered cured. These Keytruda and Opdivo and other immune checkpoint blockers are monoclonal antibodies that allow T cells to engage tumor cells, and eliminate them. But as you can see, there are only 10 to 30% of patients are responding to immune checkpoint. So my lab, as well as others in the field, are working to improve patient response rates to immune checkpoint blockers. So before going into the technical details, this is a brief background on cancer immunity cycle. When tumors are formed and when they undergo apoptosis or necrosis, their antigens are released. They are picked up by antigen-presenting cells, such as dendritic cells. And then they prime T cell responses that will, T cells will infiltrate into tumors and try to eliminate tumor cells. But with the immunosuppressive network, as well as proteins within tumor tissues, these CD T cells are turned off, leading to immune evasion. And that's where immune checkpoint blockers come in. They allow these exhausted T cells to engage and eliminate tumor cells. But as I mentioned, only about 5 to 30% of cancer patients are responding to immune checkpoint, depending on different cancer types. And these work by augmenting pre-existing immune responses. That means if the patients have cold tumor without high frequency of T cells to begin with, they wouldn't respond well to these immune checkpoint blockers. So new approaches are needed to turn cold tumors into hot tumor and do so without triggering adverse events. And over the years, we worked on many different approaches to address these problems. One of which was using smart chemotherapeutic delivery approaches to not only eliminate cancer, but trigger immune responses against the tumor cells. And we have also done these studies using photodynamic therapies. We spent the bulk of our time on developing new cancer vaccine platforms. So first part of my presentation will focus on that. And that is ideal for expanding T cells and their repertoire against the tumor antigens. And the later half of my talk, I'll talk about innate immune priming approaches, as well as microbiome modulating approaches that we've been taking lately to address these problems. First is a quick update on our Nanodisk vaccine for cancer vaccination, which is the basis for our company, Evoke Therapeutics, what the company is working on. In cancer patients, if you take out their tumor DNA and analyze them, you'll see mutations that are specific to the cancer patients. As they accumulate mutations, these tumor cells express these antigens and present them on cell membrane in complexation with MHC1 or 2. So once you prime immune responses against these what they call neoantigens, we could come up with a personalized approaches for eliminating cancer. And this approach has been demonstrated first in 2017 in different mouse models. But now in human phase 1 clinical trials, this also has been shown to be a viable approach. So what they're doing in clinical trials is isolating tumors from patients to DNA RNA sequencing. And once you find these point mutations, you can now identify or predict what antigens will be displayed on the surface of tumor cells in complexation with a major histocompatibility complex 1 or 2. Once you identify these neo antigens, you could design vaccines that go after these so that you get cytotoxic T cells against these tumor cells. So among many approaches, a peptide-based neoantigen is gaining a lot of attention, and that's what my lab has been working on. As opposed to injecting free naked peptides for neoantigen vaccination, we developed a nanoparticle system based on nanodiscs. And this has ideal properties of a neoantigen vaccine because they efficiently drain through lymphatic vessels after sub-Q injection based on their size. They're about 10 nanometers with phospholipids and very simple 20-tumor ApoA mimetic peptide sequences from ApoA lipoproteins. And onto this platform, we attach tumor antigens and as well as adjuvants to deliver them to dendritic cells. And once they are taken up by dendritic cells, they promote very potent T cell responses. And upon, in combination with immune checkpoint blockers, we see very robust efficacy. And the attractive feature of our Nanodisk platform is that this has been produced in kilogram scale before and tested in phase 1 trials in cardiovascular patients. And this has been shown to be pretty safe. So based on the proven manufacturability as well as the clinical safety of the blank Nanodisk, we are using that to deliver cancer antigens. Basically, we make liposomes in the presence of this 22-mer peptides that restrict their size to 10 nanometers. And onto this, we attach peptide antigens and incorporate adjuvants such as CpG into the particle systems. And they are pretty safe, pretty homogeneous, and they are amenable for freeze thawing and for long-term storage as well. So what we envision is making a large batch of our HDL nanodisk system, making as individual aliquots. And when patients' neoantigens are sequenced and identified, we do take each vial out, mix with our antigen and adjuvants, and have a personalized nanomedicine ready for injection. And over the years, we streamlined these processes so we can finish everything under 1 day of a procedure. So in mice, this works dramatically improves antigen delivery. So here we are looking at mouse where we do sub-Q immunizations of either free naked peptide or same peptide conjugated to the nanodisk. And as I mentioned, the free naked peptide disseminates into systemic circulation right away after sub-Q injection. So you get very little delivery to lymph nodes. But with our nanodisk, we see a very robust 28-fold increase in delivery. And we have recently shown that this is also true in non-human primates. So we also saw a 14-fold increase in non-human primates injected with our Nanodisk system. And upon immunizations in tumor-bearing mice, this results in very robust CD8 T-cell responses. So this is in tumor-bearing mice injecting a single neoantigen called the ADPGK. And after 3 rounds of vaccination, we get about 30-fold better induction of T-cells compared with one of the strongest adjuvants in clinical trials, such as a CpG and montanide mixture with a free peptide. So this gives us confidence on using our platform to induce cytotoxic T cells against tumor antigens. But in fact, in the MC38 model, with a single antigen ADPGK, in combination with our immune checkpoint blockers that are used in clinics, but this is a mouse version, we can eliminate tumors in up to 80 to 90% of cases. And this is the traditional free peptide vaccine combined with anti-PD-1, showing the superior ability of our Nanodisk to elicit CD8 T-cell responses and eliminate tumor. And the benefit of using immunotherapy is that you can elicit long-term immune responses. So if you take the survivors and give the same tumor cell re-challenge, the mice previously treated with the Nanodisk will eliminate tumor in 100% of cases, showing that this is not only great for eliminating tumors, but also preventing tumor relapse. And we have done a number of studies in different mutant tumor models, so I'll just show you the key highlights. This is the orthotopic glioma model, where we implant tumors into CNS, and then use our Nanodisk to deliver 3 different neoantigens. And this was injected sub-Q away from the tumor tissues. And after combining with anti-PD-1, we saw T cells infiltrating all the way to CNS and eliminating tumors. And we saw up to 30 to 50% of mice are eliminating tumors away. And these survivors are once again resistant to tumor re-challenge, showing establishment of immune memory response. And this is a work from Dr. Alireza Hazani, who's a crucial member of Terasaki Institute. This is his study done in non-human primates, where he attached a CM9 antigen. This is an HIV antigen in non-human primates. Because there is no tumor antigen known in monkeys, we resorted to HIV antigens. So this is showing the T-cell responses in lung tissues after 4 monthly injections in non-human primates. Here we screened the different adjuvants and identified that hiltonol is a very potent and clinically relevant antigen to combine with our Nanodisk system, where we saw very robust induction of antigen HIV-specific CD8 T cells. And what's also exciting is when we prime these monkeys and then follow with a recombinant advector boost, similar to those that are using J&J COVID-19 vaccine, what we saw was that animals that were previously immunized with our Nanodisk further expanded the CD8 T cell responses, much higher level than giving just the advector alone, showing that this Nanodisk is a promising platform to mix and match with the different vaccine technologies. So we are moving forward with these studies, and we hope to report hopefully a clinical data set in the near future. In the second part of my talk, I'd like to discuss our new approach of priming innate immune activation against cancer. And this is based on a very potent system based on C-gas sting pathway. Upon viral infection, or in the case of tumor formation, C-gas sting pathway is playing a crucial role in activating innate immune activation against tumor cells. For example, from DNA containing viruses or tumor DNA, if they accumulate in high frequency in cytosol, this is recognized by C-gas in the cytosol. And this catalyzes the reaction between GTP and ATP to form these cyclic dinucleotides, such as the cGAMP. And when cGAMP binds to sting, This activates a very potent immune activation, leading to type 1 interferon response. And why is this important? Because type 1 interferon is the key orchestrator for innate and adaptive immune activation. So in different murine tumor models, what people have shown is that if you inject these cyclic dinucleotides directly into tumor, it can have a very potent anti-tumor efficacy. And this led to a number of clinical trials headed by big pharma companies. First one was Aduro and Novartis, a compound that was injected intratumorally. Although they haven't shown good efficacy in humans, this didn't deter other companies to pursue these interesting compounds. So for example, you have BMS, GSK, as well as Merck in this field. And there are at least 12 ongoing phase 1 clinical trials using different cyclic dinucleotides for immune activation as we speak. But there are a number of limitations with these traditional STING agonists. Namely that a lot of these compounds are directly injected into tumor. And as we know, intratumoral injection is not applicable for treating metastatic cancer that kills up to 90% of patients. So we need a way to systemically deliver these STING agonists. And so far, the traditional STING agonists are not applicable for IV dosing because they are rapidly cleared, they are enzymatically unstable, and they have a very low cellular uptake because they are nucleotide-based anionic compounds. And also the safety, if you inject a lot of the STING agonist IV, this could lead to cytokine storm. So there's a safety issues as well. And also in humans, there are several human STING haplotypes. So not all humans would respond to STING agonist equally. So we need a way to universally activate STING using different compounds. And also in some cancer patients, they have a dysregulated cGAS-STING activation pathway. So that's something to address as well. So this was a project headed by a very talented graduate student, Xiaozi Sun, who discovered that by adding manganese together with the cyclic dinucleotides, we get up to 70-fold better induction of type 1 interferon response and STING activation. And as you know, manganese is a nutritional metal ion that's playing an essential role for immune regulation. And we actually take manganese from veggies. And there are several FDA-approved drugs based on manganese for MRI contrast agents. So we focused on manganese to co-formulate that into nanoparticles for immune activation. So I'll show you some of the key data set. And we initially focused on manganese because of this key paper that have shown that upon viral infection, manganese from mitochondria and Golgi are released into cytosol, activating cGAS and STING. And this prompted us to screen different metals in the presence or in the absence of STING agonist to see type 1 interferon response. So here we are using bone marrow-derived dendritic cells to look for this potent immune activator. What we show is that manganese indeed has a very interesting property of activating type 1 interferon in the absence of STING agonist. But when we add the STING agonist together, we see very potent synergy. So that's why we focused on that. And here we use the 3 different STING haplotypes that I mentioned. And these 3 cover up to 90% of all human STING haplotypes out there. So what we are showing here is that if you use the STING agonist alone, you get type 1 interferon. But if you add the manganese to that, you get very potent synergy. And in particular, these haplotypes that do not respond to STING agonist on its own start to respond by adding manganese together with the STING agonist. And manganese alone doesn't have a potent type 1 interferon activation. So all these studies were done using cGAMP, the endogenous STING agonist. And we have shown that this also works with compounds that are in clinical trials. So this is a synthetic STING agonist from Aduro. This is the one from GSK, all showing that adding manganese can significantly enhance the STING activation. And we have also confirmed this from human healthy volunteer PBMCs. So how does manganese activate STING, and why do we see such a strong synergy? The previous groups that I mentioned have shown that manganese can activate C-gas, STING. This is the upstream of the STING activation, basically showing that double-stranded DNA in cytosol will bind better to C-gas in the presence of manganese. And also the STING agonist will bind to STING in the presence of manganese to lead to good activation. In our own studies, we have shown that manganese can also act downstream of STING, activating TBK1 and P65, resulting in a very potent type 1 enantiosome formation, leading to very potent activation. And what this also means that even if patients dysregulate and downregulate STING, our approach based on manganese can still act on downstream signals to activate type 1 interferon response. So here what we set out to do was to formulate STING agonists together with the manganese so that we can achieve a systemic immune activation for treating metastatic cancer. So what we have discovered over the course of our study is that when cyclic dinucleotide, these CDNs, when they are mixed with the manganese under certain condition, they form these nanocrystals. And these nanocrystals are pretty unstable on its own, but by adding lipids with a metal chelating histidine tagged lipid, we can now stabilize them in the lipid layers. And we are using common lipids that are found in liposomes and Moderna vaccines, for example. So we have a lipid-coated nanocrystal system that has both manganese as well as a STING agonist in the core. And they are pretty homogeneous and stable with about 120 nanometer size. And as I mentioned, there are different flavors of STING agonist, and we have shown that we can make STING our particle, CMP, using different STINGs, including the bacterial STING agonist that we are using in the subsequent slides that I'll show you. So the benefit of using our cyclic dinucleotide manganese particle, CMP, is that it enhances cellular uptake. As I mentioned, the STING agonist itself is based on nucleotides that are highly anionic, so it has a poor uptake. But with our particle system, we see much better uptake by dendritic cells. And this results in a very potent immune activation with a type 1 interferon. At an equivalent concentration, our particles elicit further interferon response. So let's go into animal studies and their efficacy. So here we developed a new head and neck cancer model called NAC1 in collaboration with Dr. Leo Lei in the dental school. What he did was he generated these from smoke-induced mutation and have shown that these tumors are completely resistant to traditional immune checkpoint blockers, such as anti-PD-L1 and CTLA-4. But these immune checkpoint blocker resistant tumors respond very well when we give our CMP particles either by intratumoral or intravenous route, showing that with just the 4 injections, we can eliminate and lead to tumor regression, leading to about 80 to 100% survival. And if you inject just the free STING agonist by itself, by intratumoral or IV route, none of the animals survived. And some of these mice are showing very dramatic efficacy. Here we use the B16F10 model. This is a very resistant to immune checkpoint and traditional immunotherapies. So we wait until tumors are 1 centimeter in diameter. And without any treatments, these mice develop huge tumors within 3 days of non-treatment, getting just the PBS, showing this is in an exponential growth phase. But with our particles given at an IV route with just a single dose of injection, we see these huge tumors are sort of melting away, forming necrosis in the core. We think this has a strong immune activating potential to eliminate large tumors as well as causing tumor vasculature collapse that lead to this dramatic efficacy. And some of you may ask, if you inject a lot more free sting agonist by IV route, would that be efficacious? And the answer is no. Here in the CT26 colon carcinoma model, we give our particles by IV route, starting when tumors are about 200 millimeter cubed, or give a 5 times more dose of the sting agonist by IV dose, and that was not efficacious, showing that our particles have a significant dose-sparing ability with a good efficacy. And we set out to compare our particles to other gold standards in clinical trials. So we go back to B16F10 model, where we wait until day 9 and give our particles by IV dosing. And this was outcompeting all the other comparison groups that we had. And these comparison groups included the GSK's sting agonist that's in phase 1 clinical trial based on this paper. And we also tested it with the Argiro compound, the first-in-class sting agonist that went into phase 2 clinical trials. And we also included our control, where we changed the manganese to zinc in the same lipid formulation, but that still was far better than all the other controls. But of course, manganese was better because we saw strong synergy with the manganese with the sting agonist. And traditional liposomes without any metal was not that efficacious in this model. So this shows the benefit of having both manganese with the sting agonist in our lipid formulations for immune activation. And one of the questions people may ask is, all these tumors are spontaneous tumor model, transplantable tumor models with leaky vasculature. Is this through EPR effect? So we use the spontaneous triple negative breast cancer model, MMTV-PYMT. So these tumors, these might start multifocal tumors starting 40 days of age, and they are very challenging to treat using traditional chemo and immunotherapies. And one of the reasons is that they are poorly vascularized, and drugs and nanoparticles do not penetrate well into these tumors. So here we waited until day 75 when multiple tumors are already present in these mice. And what you can see is that injection of free sting agonist IV does not lead to any cure. But with our CMP IV treatments, we see substantially slowed tumor growth. And by day 94, you can see dramatic differences with the free sting agonist treated mice having multiple tumors all over the body, whereas our CMP treated mice are doing much better. So in terms of a biodistribution, what we are seeing is that, yes, with IV dosing, our particles are homing well to tumor tissues, much more so than injecting free sting agonist. But as you can imagine, these nanoparticles are going to RES organs, such as a spleen and liver. And we are quite excited about these results, actually, because this is all showing that particles homing to spleen may be involved in generating systemic anti-tumor immunity, and that particles may not need to home to tumor that much. Whereas in traditional chemotherapeutics, you need the direct killing effect. But if we can exploit systemic anti-tumor immunity by particles accumulating in spleen, this would be a great pathway for treating metastatic cancer. And what's also known about manganese is that it's naturally eliminated by biliary excretion in the liver. So we think particles going to liver is not posing major safety issues. And those particles that home to tumor, they are retained longer in tumor tissues, much more so than free sting agonist, which is rapidly cleared within a few hours. So this shows that even the particles, when they home to tumor, will be present for at least 24 hours shown here, and they will engage immune cells over there. In fact, within tumor tissues, these particles are taken up by dendritic cells, macrophages, and MDSCs. These are immunosuppressive phenotype MDSCs, and they activate all these immune responses. And for example, macrophages, we see high upregulation of M1 to M2 ratios. And we also saw activation of dendritic cells, CD8 T cells, and natural killer T cells within tumor microenvironment with particle treatments. And also we saw this reversal of immunosuppression as shown by decreased frequency of MDSCs within tumor tissues with our particle treatments. So overall, hopefully I was able to convince you this CMP, the particles that has both STING agonist and manganese, addresses many limitations of conventional STING agonists. We can now use these to treat tumors IV for treating metastatic cancer. And we believe this is a universal pathway to activate human STING haplotypes. So the same formulation could be used across the whole human population. And also because even if the patients downregulate SIG-STING pathway, the manganese-based approach will act downstream of a STING, therefore activating type 1 interferon response in these patients. So toward that, we are working on our large animal models as we speak. Okay, in the last part of my presentation, we want to switch gears a little bit and talk about a completely different approach for activating immune responses against cancer. Here we are taking the approach of using prebiotics, natural fibers that's existing in plants to activate gut microbiome for immune activation. And as you probably know, gut microbiome has garnered a lot of attention in the media. For example, gut microbiome has been known to promote breast cancer spread in mice. Gut microbiome also affects drug metabolism, degradation, and also even chronic pain and CNS diseases such as autism and Parkinson's disease all have been shown to be linked to dysregulated gut microbiome in patients. And a series of papers shown recently that also gut microbiome plays a crucial role in patient response to immune checkpoint blockers. Collectively, what they have shown is that when patients have dysregulated gut microbiome, they wouldn't respond well to immune checkpoint blockers. And upon the fecal microbiota transplant or transfer of beneficial microbes into these mouse systems, now these mice now start to respond better to immune checkpoint blockers. And collectively, what these studies have previously shown is that when patients, when they are not responding well to immune checkpoint, and if you take their fecal microbiota and give it to the recipient mice, these mice also don't respond well to immune checkpoint. But if you take patients who respond well to immune checkpoint and give that to these mice, then they also respond well to immune checkpoint. And what they have proposed is that there are specific bugs in the GI tract that secrete the metabolites to train T cells against the tumor tissues. So as you can imagine, there are a number of clinical trials ongoing where fecal microbiota transplant is performed to make patients respond better to immune checkpoint blockers. But as you can imagine, during this era of COVID, it's going to be very hard to make this into a drug product. So this forms the premise of our work. This is work done by a very talented postdoc in the lab, Dr. Kai Han. He screened FDA's generally recognized as a safe GRAS materials for modulating gut microbiome. And inulin, fucoidan, EGCG, and melatonin all have been shown to have a beneficial roles in gut microbiome modulation in prior studies. So we took on these and identified that in particular inulin is exerting potent gut microbiome modulating effects. And inulin, as you probably know, is a polysaccharide that's found in root plants, such as chicory root. and Jerusalem artichoke. And people are consuming inulin because it's fat as well as a sugar substituent. It has a sweet flavor, and so people are mixing it with their drink as a sugar replacement. And what we have found is that when we form this into gel and give it orally in these mice, they respond much better to immune checkpoint. So I'll show you some of the key data from our recent paper. So this is the initial screening data. So here, mice were kept on a normal chow. We didn't modify their dietary fiber. And we implant tumors away from GI tract subcu flank and give these oral candidates from FDA's GRAS materials. And then we give immune checkpoint blockers by IP route to treat tumors in a systemic manner. And this is untreated mice. This is mice treated with anti-PD-1. With the mice treatments using inulin, this is naked inulin from the powder form. We saw good synergy, and that's much better than all the other GRAS materials. So what we found was that when inulin is given, it increases these beneficial microbes that have been shown to be found in high frequency in patients who respond to immune checkpoints. So these include Akkermansia, Lactobacillus, Rosevaria, and Pneumococcus. So what you can see is that in the fecal samples in mice treated with these, with inulin, we saw higher frequency of Akkermansia, Lactobacillus, Rosevaria, Pneumococcus compared with all the other treatments. And there was a negative correlation between the frequency of these microbes and tumor size, meaning that mice that have a high frequency of microbes have a reduced tumor mass. And we think that's due to inulin that's degraded into these good metabolites, in particular short-chain fatty acids. And I'll show you some of the data in the later slides. But one of the major limitations of using native inulin powder is that it's absorbed rapidly, it's degraded in upper GI tract, whereas we want these inulin to go to colon. And that's because all these beneficial microbes that I mentioned, they reside in colon tissues. So we made a very simple hypothesis that if we make inulin into a viscous gel, we'll do that coat colon tissues better and improve their exposure to inulin. So what Kai did was he, by simple and heating and cooling cycle of inulin solution, he can make it into a gel. And with the fluorophore labeled inulin, what he showed was that after oral gavage, inulin is retained longer in colon tissues compared with injecting free naked inulin unmodified form. So this increased exposure in the colon seems to be crucial for basically providing inulin as a nutrient for these gut residing microbes. Indeed, when in the same tumor model, CT26, so these tumors are implanted away from GI tract in the subcu flank. This is a treatment using native inulin plus immune checkpoint, and our inulin gel plus immune checkpoint. And we can see that inulin gel has a far better rate of tumor elimination in up to 70% of cases compared with the immune checkpoint alone or free naked inulin plus immune checkpoint. And these are also resistant to tumor rechallenge, as I've shown in previous studies. And what's known in the literature is that mouse vendors, depending on mouse vendors, mice have a different initial microbiome to start out with. In particular, mice from Jackson Laboratories are known to respond well to immune checkpoint because they have a gut microbiome that's beneficial and responsive to immune checkpoint. So the initial studies were done in Jack's mice, but Taconic farm mice are known to have a bad microbiome to begin with, and they don't respond well to immune checkpoint. And we have basically shown that even in these mice, the immune checkpoint plus inulin gel is working well, implicating, suggesting that even if you have a different microbiome status to begin with, our inulin gel could augment immune checkpoint blockers. And we have shown this works in multiple murine tumor models and different strain of mice, including BISCNF10 in black mice. And this also works well when we combine dual immune checkpoint, anti-PD-1 and CTLA-4. Compared with an immune checkpoint alone, we see good synergy with inulin gel. And we also shown that this works in a spontaneous orthotopic colon cancer model in collaboration with Dr. Grace Chen. So by adding DSS in this APC main mice, they develop spontaneous tumors in the colon tissue. It's a good model for colorectal cancer. And what we've shown is that oral administration of inulin gel plus systemic injection of anti-PD-1 can significantly decrease tumor burden in these mice. And we have performed the DNA sequencing in the fecal samples to show that, yes, indeed, mice treated with inulin gel have a higher frequency of these beneficial microbes that I mentioned, including Akkermansia, Lactobacillus rosarioria, and this results in a good induction of immune responses. And to show that this is a gut microbiome-dependent phenomena, we depleted the gut microbiome using antibiotics. So this abrogates the efficacy. And also, if you deplete CD8 T cells, you lose the efficacy, but not when you deplete CD4s or other immune cell subsets. So this is a gut microbiome-dependent and cytotoxic CD8 T cell dependent systemic immune responses induced by inulin gel. So to further dig into this, we quantified the CD8 T cells against the tumor and have shown that in mice treated with inulin gel combo, we see higher frequency of anti-tumor T cells in blood, as well as in tumor tissues. And what was also exciting was that, in particular, tumor-infiltrating CD8 T cells have this TCF1 positive phenotype. And that's quite exciting because prior studies have shown that TCF1 positive T cells are the stem-like CD8 T cells that proliferate extensively upon anti-PD-1 treatments in mice as well as in humans, thereby providing effector cells against the tumor tissues. So we hypothesized that maybe inulin gel is turning CD8 T cells into better stem-like memory T cells. And indeed, that's the case. So here we are looking at CD8 T cell expansion in the presence of different short-chain fatty acids. So these short-chain fatty acids are what microbes produce after ingestion of inulin. And what we show is that acetate, propionate, and butyrate can further expand these stem-like CD8 T cells in vitro. And that aligns with our in vivo data set. So collectively, what we have shown is that in tumor-bearing mice, oral gavage of inulin gel plus systemic immune checkpoint blocker therapy lead to very potent systemic anti-tumor immunity. And we think the inulin gel is working by coating the colon layer, thereby providing food for gut-residing microbes. And when they consume inulin, they will produce short-chain fatty acids as well as other metabolites that train T cells to turn them into a memory CD8 T cells. And upon immune checkpoint blocker therapy, they undergo rapid, extensive proliferation, providing killer T cells against the tumor cells. And what's really exciting about this approach is that this is based on FDA's GRAS material. And this opens a wide avenue for clinical translation. And this is a quick diagram showing a number of different cancer indications that are being treated with immune checkpoint as a first, second, or third line therapy. And what we are doing is simply taking FDA's GRAS materials, making into a gel, and we believe this will lead to rapid clinical translation for improving immune checkpoint blockers. So with that, I want to thank you all for your attention. And I also want to thank all the members in my laboratory. In particular, Dr. Kai Han did all the microbiome work, and Dr. Ali Hassani, as well as Dr. Rui Kwai, worked on the Nanodisk vaccine platform. And also Xiaoqi's son is the one who worked on the CMP particles for sting activation. And also want to thank all the collaborators at University of Michigan, as well as our funding sources. So with that, I want to take any questions that you may have. Thank you.
Thank you for this fantastic talk, James. Very creative ideas. First question is, nanoparticles suffer from toxicity. Did you see any toxicity in your HDL platform?
HDL platform, there's no toxicity. We are giving very tiny dose of the nanoparticles under sub-Q route. I mentioned the prior clinical trials of a cardiovascular study with a HDL system. Even in that condition, a very huge dose of HDL was given to mobilize cholesterol and has been shown to be safe in humans. But we are taking orders of magnitude smaller dose with a sub-Q route in our vaccine approaches. So we expect it to be very safe in humans as well.
Okay, in your opinion, what are the advantages of systemic immunotherapy rather than locally delivering the agents to the tumor?
Right, so I think systemic immunotherapy is the future of immunotherapy for activating immune responses against the disseminated cancer and metastasis. I mean, you could treat local tumors with a direct tumoral injection, but in the clinical setting, that's a very tough. It's hard to pinpoint where all the disseminated tumors are, so intratumoral injection is not feasible for advanced cancer patients.
Thank you. Does inulin gel have some effect on Treg or M2 macrophages?
Yes. So we saw reduction in Tregs in tumor tissues after inulin gel treatments. And we are looking into macrophages as well.
Thank you. Can we mix 2 or 3 polysaccharides such as inulin and melatonin to get synergistic results for PD-1 treatment?
Yeah, we have an ongoing project looking into that, combining different prebiotics using inulin as the basis to further improve this. Yes.
Thank you. Have you tried loading the APD-1 into the inulin gel for a sustained release?
We haven't done that. So we are giving inulin by oral route, but you want the anti-PD-1 in a systemic circulation to go to tumor as well as immune compartments. So we could envision injecting inulin gel sub-Q, but we haven't done that yet.
Thank you. Do you have some data for delivery by your inulin gel to iliac or mesenteric nodes?
We are working on that as we speak, yes. So we are looking into uptake of inulin and other components in the GI tract draining lymph nodes.
Thank you. Regarding these manganese particles, do you think the cells, they enhance the uptake, but is it the surface effect? Is it chemistry effect? Is there a dose limitation? It's very creative and novel. Can you elaborate a little bit more? I mean, you showed a slide where you compared other metals, zinc and iron and other things. How does this one stand out and why?
Right. So we screened the different metal ions and saw that manganese is in particular very good at activating type 1 on its own, but in the presence of sting agonists, we saw very potent synergy. We think manganese is involved in, it's acting as a cofactor in some of the enzymes involved in C-gas-sting pathway. We also saw cobalt plus sting agonist has a strong synergy, not as strong as manganese, but cobalt also had this synergy. But we focused on manganese because of prior FDA-approved our MRI contrast agent that has manganese in its formulation.
Thank you. How would systemically mounted responses generated by CDN manganese particles uptaken by spleen immune cells specifically target tumors versus other circulating pathogens?
Yeah, as we showed in the biodistribution, we see them CMP taken in the spleen tissues, and APCs over there are uptaking them, as well as APCs and immune cells in the tumor tissues. Yeah.
The second part of the question, if the response is non-specific or multi-specific, is there a limit on tumor size burden that you have seen that would allow for this mechanism to control tumors before immune-related adverse effects occur?
Yeah, we are doing those titration studies using these CMP, and we also currently doing rabbit studies. There's a good rabbit tumor model based on VX2, and we are trying to scale up the production and ensure efficacy in larger animals.
Thank you. What effects does inulin directly have on gut microbiome? Does it increase a certain bacteria that help the patient's immune response?
Yeah, so inulin is a food source for microbes, and our mammalian enzyme do not cleave inulin. So it's only cleaved by bacterial enzymes and serving as a food source. So once they digest it, ferment it, they produce these metabolites that I mentioned, short-chain fatty acids, and they are the key drivers for improving immunity.
Thank you. Can we use insulin or magnesium with small peptide replacement of immunotherapy?
Could you repeat the question?
Can you use insulin or magnesium with small peptide replacement of immunotherapy?
I think that could be possible. Yeah, combining peptides with other approaches. Yeah.
Thank you. So my final question, is there any benefits of mixing inulin with whey protein?
Whey protein.
Yeah.
I'm not sure. Yeah, we haven't done that. That's something to look into. Yeah.
Well, thank you so much for this very creative, very fantastic talk, James. I appreciate your time.
All right. Thank you very much, everybody. Thank you.
Have a nice day, sir.
Okay. Bye.